So we have Siemens from the Heidelberg University in Germany, and the talk is how to cope with heat. Neuronal plasticity can help.
Good. Yeah, thank you very much for the introduction and also for having me here. I'd like to thank the Kavli Foundation and Alan for putting this very exciting and diverse meeting together. So my background is actually sensory physiology. I worked a lot on trip channels, TRP channels, and how they help to sense temperatures. But for this talk, I really would like to focus on one subject that, or one project that really excited us, and that is related to this title here from a New York Times article last year, how our bodies can adapt to heat. And if you look at the fine print, that can happen in a few weeks in mice that we use as model organism, still among others by now. That takes also 3 to 4 weeks to do. And my hypothesis is, a key also from our study, is that neuronal plasticity is a key driver for this type of heat acclimation. So where would we look for such plasticity? So let me give you a little bit of background. In vertebrates, mammals, the preoptic area in the hypothalamus is sort of considered, in a very simplistic view, as a thermostat. It regulates body temperature, and to do so, it integrates temperature information from peripheral and central sites. Now, over the last years, several groups, including us, have contributed to dissect what cell types in the preoptic area are helping or contributing to this. In particular, people, including us, have focused or found so-called warm responsive cells, or neurons activated by heat. So what would happen if you activate those cells? So this is just depicted for time reasons in this cartoon. So if a mouse or an animal is faced with heat, it is in danger of hypothermia that triggers activation of these warm activated neurons, and that in turn then triggers cooling responses. So a classic to prevent sort of the overheating and to bring back the normal steady state. So a classic acute heat defense homeostatic behavior or physiological response. So, but what happens during long-term stimulation, long-term heat stimulation? You cannot acutely defend this. It would wear the body down. So you have to do, or the organisms, and most organisms, maybe all organisms, can do this to some degree, adapt. What I like to call rheostasis. So you shift thresholds of physiological parameters to make it work, to adapt to this changed environment. And the key question for us is the POA, the thermostat region, sort of involved, and if so, how? Now, let's look at what adaptation and heat acclimation really means. It means a lot of things, and that has been studied quite extensively in the periphery. So blood flow changes. Brown fat completely the mice get rid of. They eat less, they lose weight, fluid balance changes, and even behavior changes. So adaptation to hot environments for this mammal changes a lot of different things. And that, like I said, in classic physiology books, it takes about 3 to 4 weeks. Not everything, but particularly the metabolic changes take quite some time. So this all helps to maintain a stable core temperature and prevents overheating, and that we can measure. So Sarah, gifted postdoc in the lab, previous postdoc, she developed or modified this acclimation assay. So this is how it really looks. It's a sort of repurposed incubator where we put in light and telemetry to measure the body temperature of these mice. We acclimatize them for different times, and then expose them to a heat endurance assay while measuring their body temperature. And this is what such an assay looks. So you can see here the body temperature over time in this heat challenge assay. And in mice that are not acclimatized to heat, they reach this cutoff temperature here, 41.5, fairly quickly, in a few hours. So we take them out from this assay then. But mice that have been acclimatized, they can endure much longer, even beyond the 24-hour period. And this is what we can quantify in this, what we call endurance time. Now, so mice exposed to heat in a controlled way for a long time can gain heat tolerance. So we have a physiological assay that we can now probe and ask if the hypothalamus, the preoptic area, is involved. And why should it be involved? So we have a very simple hypothesis here. Strong, long-lasting stimulus, classically, can result in plasticity. So we reasoned that the strong, long-lasting heat stimulus transferred via the peripheral nervous system to the preoptic area would potentially trigger some form of plasticity in these warm activated cells. So to study this, we used mice that have a genetic label, a GFP, in these neurons that we and others described before. And Wojciech, physiologist, he then exposed mice to different temperatures for long periods of time, and then performed whole cell patch clamp recordings in slices obtained from these mice. And the first results were quite humbling. There was not classic synaptic plasticity, or at least not much. But what he found was that these neurons become very, very active. So you can see here an example of a slice from a control mouse. Many of them are actually silent. They don't fire at all. But if you take slices from mice that are acclimatized to different times, they become very, very active. So heat acclimation increases tonic firing of these warm activated cells. So that is quantified here. As you can see, I've lost my cursor, but I think it's very clear. So the non-acclimatized are fairly low active, and then they become very, very active. So this cell population is just shown here. It's a very discrete population around the third ventricle, so the early part of the anterior hypothalamus. Now, one feature that struck us even more was that they don't only become tonically active as shown here, but that the cells become themselves warm sensitive. And that is shown here. So on this side here, you can see if we expose in the bath solution the slice to higher temperatures, they become very, very regular and temperature sensitive cells, which we also quantified, and this was strikingly significant. So, and that was in the presence of synaptic blockers, so it's really a cell autonomous function. So they become warm sensitive pacemakers. So here's just a quantification. It was strikingly almost 90% of these cells turned to those warm sensitive, transformed into these warm sensitive cells. So we also looked at slices is good and nice, but what about in vivo? So we used miniscope imaging of GCAMP positive neurons using the mouse genetics. And to cut a long story short, here you can see in this simple experiment here, so this is pre-acclimation. We stimulate these mice with warm temperatures, and you can see already there's a few cells that light up dimly, but after heat acclimation, not only more cells light up, but they light up much, much stronger. So the warm responsive cells also increase dramatically in vivo. So really saying that ex vivo and in vivo, these cells transform to become highly temperature sensitive and tonically firing. So what I've shown you so far, we can heat acclimation transforms cells to become tonically active, a selective group of preoptic neurons. And what I also showed you before, that one can assay this physiologically, that mice can become more heat tolerant over time. And the million-dollar question at the time was, is that causally linked? Is the changed firing properties driving this change in physiology? And to look at this, we used the wild or the broad tool set of neurophysiology. In this case, we inhibited these neurons with a so-called GI DREAD, an inhibitory receptor that we artificially, with a virus, introduced into those cells. We first tested that this system also works. As you can see here in this electrophysiological experiment, if we give the drug CNO that activates this receptor, we can silence these cells very effectively, as shown here. So then we were ready to test in vivo if the activity of these neurons matter. And for that, we first, in a control experiment, non-acclimatized animals, exposed these animals to heat in this heat challenge assay, and then injected CNO to block activity of these neurons. And here's the result. Each curve is the core temperature, the body temperature of a mouse. In red, the control mice, and in blue, the ones that we inhibited. And there's not much difference. So shown here in this quantification, the baseline heat tolerance is not changed if we block these the cell activity. Maybe not so surprising, as I showed you in the slices, many of these cells are silent and not very active. So if you block something that is not very active, not much expected there. But then when we acclimatized these animals first and repeated the experiment at the end of the acclimation in the heat endurance assay, this was a shift of night and day. So the acclimatized control animals really were heat tolerant, and the acclimatized but then inhibited mice failed very early on. So if you take one thing from this talk, so really an acute heat challenge, these neurons seem for the heat tolerance not to play much of a role. But once they have transformed over this long time frame in the heat, they become really a dominant factor driving heat tolerance. And if you block this, the mice do even worse than before. So somehow the capacity to mediate heat tolerance has dramatically increased. So with this, I think I hopefully have still a few minutes, but this is the summary of this. So what I showed you is, and I, for time reasons, couldn't go into all the details, signals from the peripheral nervous system converge temperature information onto the POA, and if you do that long enough and strong enough, that transforms these cells plastically. This is all reversible. We tested this. It takes about a week to go back to normal into these highly tonically warm-sensitive neurons, and that somehow, we don't know all the parts that happen then in the periphery, convey heat tolerance to mice. And here, a last-minute slide. We were asked as speakers to see what are gaps and opportunities here. I think one aspect is to see if this is something that is conserved among other vertebrates and non-vertebrates. Particularly in this meeting here, I would very much like to see if in other species one finds some of those acclimation neurons as well. Would plasticity be different in a natural environment? We talked with some of the members of the audience here about this. This is a very artificial system we use. We only change temperature, we keep humidity in place. But how would this look if one does it in a more naturalistic setting? Would the plasticity be also there? Would it be modified by other parameters, for example, fluid availability and things like that, if one thinks of a desert region? What are the downstream targets? What happens? And also, if one thinks about the beautiful talk from Yves Mader this morning, what happens in the circuit? Is there changes in the input-output relations of these neurons? And can the plasticity be harnessed for medical purposes? So we measure the mice lose weight. For example, can one use this to sort of understand mechanisms, how this plasticity might be helping obesity, for example, as one example. And then what it needs to do these things? Of course, academic freedom. I had to put this here at this meeting, and funding as well. And with this, I'd like to thank you and happy to answer questions.
We can start with Kira in purple.
Hi, great talk. I was wondering if you guys plan to or had yet done the opposite of the inhibition and stimulated these worm-sensitive neurons in the POA in non-acclimated animals and see if it changes their sensitivity.
This could be almost a stage question. I have one more slide right after this, so it is a stage question. The postdoc who did this, he was also, can we do the opposite? So this is the experimental setting that I guess you had in mind. So we stimulated with channel rhodopsin to have a really good control by light these neurons. We used a very low frequency because these neurons acutely mediate heat defense. That if you stimulate them strongly, the mice will go into hypothermia. That is actually a nice way to control that you hit the right region. And so then we challenged them with heat in the endurance assay and asked if activating these neurons without the animals ever having been exposed to heat before would convey some extra heat tolerance. If you do this just for a day, so 24 hours, it's not enough. The mice fail quite early, as you can see here. If you do this for 2 days, you start maybe to see some separation. But if you do it for 3 days and longer, indeed, the mice gain heat tolerance. So here, this assay is already cut off for practical reasons after 9 hours, so a normal working day. I don't know how much longer they would do, and if one can push this even further. So what I like about this, the complex aspect here is probably these neurons do 2 things. They slowly increase their firing, slowly priming the body and different organ systems to be ready. And then the increased warmth sensitivity, so that's the first experiment. We block them at the very end, so while everything has been primed already. So they must have some acute function that is very sensible now. And what organ systems, my best guess would be cardiovascular, probably some metabolic aspects, maybe even liquid fluid handling is acutely then sort of blocked, and therefore they fail within minutes if you block the activity at the end. So 2 different functions. The slow development, it's our hypothesis, the slow development of gaining heat tolerance and then executing sort of heat tolerance mechanisms at the end when you are exposed to an extreme heat event. Yeah. Thanks for the question.
Hi.
We can continue some questions.
So to answer the first question of gaps, I think Hydra might be a place to look. So they have a population of neurons that fire tonically and rate increases with temperature. And their ability to survive high temperatures requires these pre-acclimatization steps. So we can chat later if you want.
Yes, let's please do. Let's have a chat afterwards. Yeah.
Yeah.
Sorry if I missed this, but how exactly does the neuron start firing tonically? Do you know if there are more trip channels that are expressed or some other kinds of channels that allow them to switch from this quiescence to sudden tonic firing?
Yeah. It's another almost stage question. I mean, I come from physiology and ion channel research. I maybe can see if I pull this up. So trip channels was the first thing. I have been trained in David Julius's lab, so as a postdoc, working on various trip channels. That was actually surprisingly not much going on. But what we found is, let me just show this here. So the biggest, that's your question essentially, right? What transforms these cells to become highly regular and temperature sensitive? So Wojciech, he looked at all kinds of currents. This had to be in slices, so isolating currents is tricky, as many physiologists would probably agree with. But he managed to get, in these nucleated patches, this was the biggest change in current that we found, a clear sodium current. For pacemaking, that makes sense. So this channel we found is actually increasing what is called a persistent current, which at a low pace, so this is not like bursting or very high rates of firing. This is 10, 20 hertz. And this persistent current is probably driving it. We sequenced these cells, we FACS sorted them and sequenced them, because by itself, enough channel probably cannot do all of it. And we found a couple of potassium channels changed as well. That doesn't answer your question, what is the heat sensor here? I mean, it could be based on also what we learned this morning from Yves Mader, multiple things, right? I mean, that tiny changes in conductances based on temperature that in this way make the firing very temperature sensitive. I'm not saying that trip channels is not involved. We didn't go systematically through all of them. But as a main current, it seems like that drives this is a sodium current. That's at least our current knowledge.
Shall I go next?
Yeah. Can I ask a question too? Yeah. So I was wondering if you test the opposite, like you do the hot acclimation, and then you come back to cold acclimation, and you observe that the neuron became silent.
So it takes a while. So everything here is on a slow pace. That's why I also, it's somewhat unique type of plasticity. If one thinks about classic synaptic plasticity, you need tetanic stimulation for a few minutes, an hour maybe. This, we don't see anything within 24 hours. And the opposite, if you de-acclimatize them, the activity seems to be higher still for a few days. We have never tested the warm sensitivity because if that goes always hand in hand with the tonic activity, so we don't know. And it goes back down to baseline after 5-6 days, plus minus. Mm-hmm. It's a bit tricky to do this experiment. The mice are not very happy. If you acclimatize them to heat, they've shifted, right? They now are in heat, and if you put them in cold, they cannot keep their core temperature. They will drop down, so you have to, if you do it in a friendly way, do it successively over several days with slowly decreasing temperatures. But yeah, it takes a few days to come back down.
Yeah.
And it takes even longer than for them to grow back their brown fat, to be able to, I mean, we keep mice classically at cool temperatures, right? Our labs is a subtle cold challenge for mouse.
I think it's super interesting because in the context of climate change, we are going to face like very fast variation of temperature. And so like what you say is like maybe the fact that we are going to like switch so fast between different like temperature conditions, like we might not, I mean, they're mice, not human, but we are might not be able to adapt or resist at this fast fluctuation.
I would love really to do this experiment to mimic a more sort of variation type of thing. Also, we keep our mice always constant, right? At least in our vivariums. So with this incubator, you can program it to be hot over the day and then cooler over the night. And even then become more naturalistic by giving them burrows to crawl away and to see how the plasticity change. Yeah, lots of things to do. Okay.
Thank you so much.
Yeah.